Hello, everyone. Um, we have already finished the top-down control of zooplankton dynamics. What do you understand by that? What are the different attributes to it? Now we are going to explain it with the help of an experiment that was performed a way back. So before going in details with this experiment, that how the top predators in a particular ecosystem influences the zooplankton community, at the low trophic levels, we'll just see uh, who was the first limnologist that explained this phenomena. So Herbeck in 1958, he was the first one who demonstrated that if we manipulate the planktivorous fish number and abundance, it will have a dramatic cascading effect on the zooplankton abundance. And that is what have been exploited in this experiment. So this experiment, if you see these two graphs, so this experiment it was done in a lake called Crystal Lake, Connecticut, in U.S. So in this particular lake, the data of zooplankton community it has been sampled in 2 years, one in the 1942 and the other one in 1964. And what was a major difference with respect to the predator is that in 1942 there was no planktivorous fish present in the lake. However, in 1964, a fish named Elusa estivalis, it has been introduced into the lake, and then the samples, the plankton samples for both the times, they have been analyzed with respect to the zooplankton abundance, zooplankton species, and also one important parameter is the body size of the plankton, irrespective of the species. All the zooplankton, they have been categorized based on their body size. So this is what this frequency distribution histogram has represented. So if you look, let's go one by one. So first of all, let's see what this particular graph from 1942 it depicts. So in this graph, if you see this is the x-axis, it's the length of the body. So it ranges from 0.2 mm to it goes up to 5 mm in case of Lepidoptera. So this is mostly or most of them these belong to the crustacean zooplankton we are talking about here. And this on the y-axis, it represents the frequency distribution. So that's what this histogram is. So in this, if you look at this particular graph, it is very clear that maximum percentage of the zooplankton, it lies with the group that has a body size of around zero point 0.8 mm. And then maybe zero point 0.7, if you see this is a zero point 0.7, 0.6. So there is a peak or trend you can see that the point from zero point 0.6 to zero point 0.8 it raises and then it goes back to like 1. So this represents that when the this particular zooplanktivorous fish was absent in the lake, a large number of large-bodied species of plankton, of crustaceans, they were present, and that included diatmus. Then we have cyclops, Daphnia, mesocyclops, Lepidoptera, Apishura. So all these are dominant zooplanktons at that particular point of time. Now, what happened when the fish, zooplanktivorous fish, has been introduced after 1964? So, when the fish was present, we know that this particular fish it feeds on zooplankton, and because it's a visual predator, it feeds on the large-bodied zooplankton. So, it controls the population of the next trophic level, which is zooplankton, and thereby it changes the community structure of the zooplankton population. By what? That most of the species now present once the fish was introduced belong to the group where the body size it ranges from zero point 0.2 to zero point 0.6 only, and that too maximum percentage is from zero point 0.3 mm in body size. So now the shift from the species which were dominant in this particular 1942 year, now the shift is towards the small body sized crustaceans like Bosmina, Tropocyclops, Cereodaphnia. So these are all small bodied zooplankton. So this clearly indicates how the top down control works in an aquatic ecosystem with respect to zooplankton population. So whenever we change or we manipulate top predators, it will show a cascading effect on the population of the trophic levels below it. So um, I hope this is clear. In case of any questions, feel free to reach out to me. Now we are going to move on to our next slide.